So I'm going to talk about hair cell depolarization first, and that is because with the ear, both the balancing portion and the auditory portion, all of it operates on hair cells. And so if you don't know how hair cells do what they do, it's going to be really hard to understand the rest of what I'm talking about. So for that reason, hair cells are a good place to start. So I'm going to draw a really, really big hair cell. And then actually, in order to do that, I'm going to move my label over a little bit so I have extra room to draw. So hair cells, it's a bit of a misnomer, including the name for these things I'm about to draw. So the tall one at the left is called the kinocilium, and I'll label this once I'm done drawing. So if you're, sort of scrambling to write that down, don't worry about it. I will do it for you momentarily. And then let's give our cell a nucleus. Perfect. And I'll start labeling. So this is called the kinocilium. And the reason it's a misnomer, which means wrong name, is because this is not cilia. Cilia are long filamentous hairy structures that have a microtubule array in the center of them. The kinocilium is actually a specialized microvillus. So the reason that they have that weird wrong name is just because at the time these were discovered, we didn't have the microscopic resolving power to tell that they weren't cilia. So they were like, oh, they look like cilia, let's call them that. And then it turns out later discovery, they're not actually cilia, but the name stuck. So that's what I mean. So for every hair cell, there's one kinocilium, and this is the tallest one. And then these little guys here are called Stereocilia. Again, not actually cilia, but that's what they're named. So now that I've labeled those, and I'm just going to make an arrow so I have room to draw here. The other thing you need to know is that all of these little specialized microvilli are connected, and they're connected via a very complex and special protein. So there's a little protein hatch in the tip of each of these Stereocilia. And that hatch is connected to the tip of the neighboring cilium by a protein that looks a lot like titin. You might remember titin from the sarcomere. It helps to stabilize the sarcomere. This protein is similar. It's sort of a spring-shaped protein that connects the little hatch on one stereocilium to the tip of the neighboring stereocilium. So we call this, it's creatively named Tiplink protein. Imagine that. Okay, so the other thing I have to draw in here is vesicles of neurotransmitter. So receptor cells, like hair cells, they don't generate action potentials in their own surface. They instead generate something called a receptor potential, which is a depolarization of the cell that's being stimulated enough to exocytose neurotransmitter onto a neuron. So I need to draw that neuron as well. So here we have a dendrite for a nerve cell, and it's going to go off and join the rest of whatever nerve it's part of. So the other thing you might have forgotten from the eye and ear stuff, or the sensory systems in general, is the fact that the inner labyrinth of the ear canal is filled with a specialized fluid called endolymph. So I'm going to sort of make a little container here. And I've already labeled things, so I'm going to erase just so that I have room to draw all kinds of stuff now that we've established our labels. And I'll refer back to the words that I just erased, so we'll have reminders of which thing I mean, but right now I need to draw some stuff. So outside of whatever membrane is enclosing the thing, and the reason I'm being vague about that is because what membrane that is and where it's located depends on where you are in the inner ear. So we'll get to that later, but this is called perilymph. And perilymph is similar to plasma. Endolymph is different. So outside in the perilymph, it's similar to plasma. And if you think about cells in your body, there are many, many different kinds of cells that spend a lot of time pumping sodium out and pumping potassium in using the sodium potassium ATPase. And what that ends up meaning is that in most of your body, the extracellular fluid is high in sodium and low in potassium. So let's write that. Now here's where it gets weird. In the endolymph, it's the opposite. There's lots and lots of potassium in the endolymph and relatively little sodium. So if you were thinking about, like, remember when you had to learn the histology of the cochlea, for example, and you had to learn that the central chamber that contains the organ of Corti was filled with endolymph, and you might have wondered to yourself, why do I have to remember what's in here? Why is that important? This is why it's important, because endolymph is a very peculiar fluid. It does not reflect the ion composition of 99% of the rest of your body. It's a unique and specialized fluid. So if I tell you that this little green guy is a hatch, meaning it's a protein in the membrane that can open, what's going to happen if I push the stereocilium that way?
Potassium's going to go in.
Yeah, exactly. So if I push the stereocilium in the direction of my arrow, and let me draw that. So now I've pushed it over. And actually, why don't I just go ahead and draw what happens when you excite a hair cell in general, because it's worth it. So we have the little hatches that have been pulled open because the Tiplink protein is being stretched because of the force pushing the kinocilium over, which you were correct, means that potassium is going to enter the hair cell. So it is depolarizing, the hair cell is, because it's receiving an influx of positive charge. It's just not depolarizing in the manner that you guys are used to, because you're used to thinking about action potentials that are sodium mediated, not potassium. So the receptor potential here for the hair cell is potassium based. And that produces a depolarization that spreads throughout the cell's surface. And when it gets to the bottom of the cell, that depolarization wave hits some voltage-gated calcium channels, which let calcium in, and that causes fusion of these vesicles with the membrane, which lets neurotransmitter out into the synaptic cleft so it can bind to and excite this neuron. And this neurotransmitter is glutamate. So if you push a hair cell's cilia, I'm using that in air quotes, over, if you push the kinocilium away from the stereocilia, that generates a receptor potential. If you push the kinocilium in the opposite direction, that's inhibitory. So before I move on to other parts of the ear, do we have any more questions about how hair cells depolarize? Okay, so that's one hair cell. You have many millions of hair cells. And here's the cool thing about hair cells. They can detect different stimuli that feel different to you. So you perceive the stimulus differently, even though at the end of the day, it's just what's happening in this picture, happening many times in a row. So let's start with something relatively simple. And that was going to be the crista ampullaris. So to give you an idea of where I'm talking about, I'm going to draw a small representation of the gross anatomy of this stuff in the corner. And how shall I put this? This is a hard thing to draw and it might look really, really ugly. So I apologize in advance if it is. Okay, so each of these is one of the semicircular canals. And then this little widened area is the ampulla. So ampulla is just kind of a generalized term for a widened area that is attached to some sort of tubular passage. So this is the ampulla of the semicircular canal specifically. And inside of here, there's a little ridge of cells, and that's the sensory apparatus. So what I'm going to do is look inside the ampulla. Oops. So what I'm going to do is draw you the cross-sectional view of the face of this mound, but really it's a little crest. So it's more like a raised hill that is continuing on. It's just this face right here is the cut face of it. So that's what I'm going to show you. So there's some kinocilia and stereocilia. There's our hair cells in completion. And then here is the axons that are innervating those, and they're going to go off and converge with other axons to form the vestibular branch of cranial nerve 8, which you might remember is the vestibulocochlear nerve. So again, these are surrounded with endolymph. And this apparatus is cool, but it's not by itself enough to give you the sensation of acceleration. So let's add the necessary component, which is the cupula, which is kind of a gelatinous mass that sits on top of the crista ampullaris. And in the gelatinous mass is embedded the kinocilia. And then just to keep us, remembering that this is not an isolated structure, I'm going to draw this kind of going back, going back further. So we have hair cells all along this crest, which I'm going to draw quickly and sort of badly. And so too, the cupula is going to extend backwards. So all of these little kinocilia back here are also embedded in the gel of the cupula. So as you can see, based on the way I've positioned these kinocilia, if this gel gets moved this way, we're going to have excitation of this vestibular branch of cranial nerve 8. And if the gel gets moved that way, I'm going to have inhibition of these particular neurons. So the easiest way to understand this is think about, and I'm going to ask you to really change what your mind is on for just a moment, but I promise it'll be worth it. Let's say I have a ribbon. Oh no. There we go. Let's say I have a ribbon and it's attached to a stick. And while I'm not moving, it's just kind of drooping. What happens to this ribbon if I start rapidly running that way? What's the ribbon going to do?
Stick up, like go straight?
Yeah, it's going to stream behind. So it'll move from this position to more of like a flapping behind me position, right? So that's the way the cupula works. The cupula is more dense than endolymph. And it behaves like the ribbon. So if, let's say, the person who possesses this crista ampullaris, if this person moves this way, the cupula is going to tip in the opposite direction, exciting the hair cells. So the way that that feels like angular acceleration, like motion to us, is if you look carefully at the way the semicircular canals are oriented. They are oriented with an X, Y, and a Z plane. So the orientation of the semicircular canals, each with their own ampulla, gives you vertical motion, horizontal motion, and oblique motion, because you have 1, 2, 3, one for each of the spatial dimensions. And you have one of these in each of those spatial dimensional semicircular canals. So if you're still having a hard time understanding this, I completely understand, because it's a really complicated thing to try and, sort of mentally conceptualize. But I'm going to give you some examples of times when you might have actually experienced angular acceleration that might help you understand a little bit better. So the first is, when you are in a car and either you or the driver accelerate around a curve too fast, and you have that feeling of going around a curve really, really quickly. So it's not only in your body, but it's also in your head as well. That is the angular acceleration. So angular acceleration is acceleration around a curve. When you have that feeling when you're driving or in a car, what you're detecting is your cupula in the crista ampullaris associated with horizontal plane, detecting angular acceleration to the left or to the right. Or if you're on one of those rides at the carnival, like the ones that are fashioned after Viking ships, where they go, swing back and forth until you're almost vertical, that's going to be detected by this one. The motion of a roller coaster would be intermittently detected by all three, because roller coasters have you accelerating around all kinds of curves. But really what it all comes down to at the end of the day is this dense gel is moving opposite the direction that you're accelerating. So if I'm going in the direction of the red arrow, the cupula goes in the opposite direction. And that gets turned into action potentials in the vestibular branch. And that's sent off for processing by the brain. And that's how you perceive depolarization of these hair cells as the sensation of angular movement in three-dimensional space. So that's the crista ampullaris. Let's look at the macula. So the macula saculi and macula utriculi. Macula means spot in Latin. So macula saculi means spot in the saccule, and macula utriculi means spot in the utricle. So the utricle and the saccule are attached to the semicircular canals, and they also contain hair cells, but their hair cells are arranged differently. So it's actually a little bit simpler even. So let me draw myself some hair cells here. They're all embedded in a plane. And then in this case, the gel is just right over the top of them. So there's a gelatinous mass just sitting on this plane, and embedded in it are little rocks. And I'm not kidding. They are little crystalline pebbles, basically, of calcium and phosphate and other minerals that are embedded in the gel. So these have a name. Otoliths, which means ear stones, also called otoconia. So this structure works on the following principle. As long as my head is level, so parallel to the ground, I have no excitation of my hair cells. But if I tip my head, let's say I tip it this way, the otoliths are heavier than the gel, and so they're going to pull the gel in the direction of gravity. So if I tip my head back, the otoliths are going to pull the gel in the direction of gravity, so towards the ground, and that's going to move my kinocilia away from my stereocilia and tell my nervous system that my head position has changed. Which again is going to be computed by the vestibular branch of cranial nerve 8. So you can even experiment with this on your own. So if you close your eyes and tip your head forward, you'll find that even though your eyes are closed and you don't have any visual cues about the position of your head, you can definitely tell that your head is not level. It's tipped forward. Same deal with tipping your head back or side to side. So the macula utriculi and macula saculi, their power is in telling you when your head position has changed in relationship to the axis of gravity. So this gives you your sense of up and down. So to help you understand this a little bit better, we can use some other real life examples that most people are familiar with. So think about when you got into an elevator, and I think we've all been in elevators that maybe accelerated and decelerated a little bit too fast. You tend to find these in really tall buildings so that the elevator is not slow, but they can make you feel nauseated. So that lifty feeling you get when the elevator is going down a little too quickly, that's computed by this structure. Same deal with the heavy feeling you get when the elevator is accelerating upward a little bit too quickly. That's the feeling you're getting because you're moving upward against gravity and the otoconia are resisting that and pushing down. And this is also accounting for linear acceleration, not only vertically, but horizontally. So, if your eyes are closed and you're in a car, you can tell if you're going fast, and you can especially tell if the driver is speeding up. You get that feeling in your head and your body. That's computed by this structure as well. But again, all of this is just clever ways to arrange hair cells that give you different feelings and different perceptions of feelings based on how the hair cells are arranged, but they're all at the end of the day letting potassium in, generating a receptor potential, and exciting the vestibular branch of cranial nerve 8. So that's how your ear works, which is pretty cool.